his laptop and the device and jump into a tuk-tuk or a taxi. And what we did was a few experiments, mainly whole genome shotgun sequencing to start off with. One day when we walked into the lab, there was a patient who had just walked in with a fever, and we took a blood sample from him. He was diagnosed by microscopy as having malaria, and we took a larger blood volume, and right there and then we extracted the DNA and we ran it on the MinION device. So the same day that that patient was diagnosed, we were able to sequence the parasite's DNA, and that was really groundbreaking. And then on this particular trip, we've changed our protocol. So we thought, well, instead of sequencing the whole genome of the parasite, why don't we try and sequence just selected regions of the genome and do that by amplicon sequencing. So in a previous study, we'd already designed primers to amplify up these drug resistance genes in Plasmodium falciparum. And so Zheng Ping brought those primers with him, and with 7 or so pre-existing patient samples, he did the PCRs, amplified up those potentially drug-resistant genes, made libraries, and was able to run them on the MinION device. But at the same time, once again, we had 2 patients that walked into the clinic, and on the very same day that they were diagnosed with malaria, we got sufficient blood to amplify up their potentially drug-resistant genes to see if they may be harboring resistance alleles to the antimalarial drugs that they were given. So this for us is very exciting, because basically what it means is that we can now work towards personalized medicine to some extent, tweaking the drug treatment that is given to that particular patient. It's very exciting for us, and has really revolutionized the way that we are doing our research.